Hear me?
Yes, we can hear you.
Thank you. Let me see, what do I have here? Let me share my screen and we will begin. Should be seeing my screen now. Let me make sure I'm recording this. Yep. All right, today is September 28th, and today we're going to talk about Lab 01, the ubiquity of microorganisms. Let me remind you that Lab 00, the worksheet must be turned in by 8 a.m. this Friday. And then Lab 01, the worksheet should be turned in by 11:59 p.m. this Saturday. Also by 11:59 p.m., you should have the online plagiarism quiz taken. And don't wait to the end for the plagiarism quiz, because if you don't get 100%, you can retake the quiz, something up to like 7 times. Let me remind you that you should read the infectious disease research project file on Canvas. Let me see if I can find that. I'm not in Canvas. Does anybody know why they removed Zoom from Canvas? I don't remember receiving an email about that. Well, this doesn't seem like it's working. Oh, it's just slow. Oh, I know why it's slow. Because the lecture files are doing something from the Zoom session, from the lecture, and so my computer is going to be slow. All right, there's the infectious disease research project. You should at least look that over, if not read it by next week, because I'm going to talk about the infectious disease research project next week. And when am I talking about that? It looks like we'll be talking it on Monday. Not Monday, Tuesday. All right, any questions about what we're doing? If not, let's go to Module 1, Ubiquity of Microorganisms. For most of these labs, there will be some reading that you should do before doing the lab. So there's reading about culture media on page 157 to 162, and then obtaining pure cultures on page 162 to 163. So not very much reading. There will be some supplemental material like video clips in this lab module. The learning objectives. Upon successful completion of this lab, students should be able to know that microorganisms are almost everywhere, be able to describe the purpose of culture media, and describe what colony abundance is and colony morphology. You should also be able to define the terms used in this lab. Any question about any of that? All right. Oh, come on, where's my mouse? There it is. Microorganisms are organisms that are too small to be seen with the naked eye, meaning an eye without a microscope. I'm just going in and highlighting this because I should have this highlighted and I don't have it. Viruses and prions are included in this group, even though they aren't really alive, and it's debatable whether viruses are alive or not. We'll also include helminths, worms, even though the adults are not microscopic. However, their infective stages are. The phrase ubiquity of microorganisms refers to the fact that microorganisms are essentially everywhere. It's not entirely true. You can find areas on the planet that don't have microbes, but that would be really lava, something like that. In our everyday common experiences, microbes are virtually ubiquitous. They're in the air, they're on the food we eat, they're in the water we drink. They contaminate surfaces everywhere, as well as our skin of our fingers. The only place where microbes are not found would be internal to a human body or maybe an animal body, like in the blood, the cerebrospinal fluid, the muscle. Normally, those areas do not have microbes, and you would have an infection if you did have microbes in those areas. We invest a lot of energy with our immune system to keep these fluids and tissues sterile. That means free of living cells. I should say, that's not true of our cells, but free of contaminating microbes. And we get sick when our immune defenses fail. In this lab, we're going to investigate the abundance and diversity of microorganisms in common environments. We'll also learn some basic lab protocols. Any question about any of that? So in microbiology, we will give you media which is sterile, meaning it has no living cells in it. And we call that a culture medium or a growth medium because we grow microbes in it. How we sterilize it is we usually put it in an autoclave, and that would kill all living cells, even endospores. A culture media can be liquid, we call these broths, or it can be a solid by the addition of a solidifying agent such as agar. We'll talk about agar a little bit more later. All right, we have a little video for you to watch preparing pour plates. You should note how culture media needs to be sterilized before use. An important question to consider is, why must a culture media be sterile prior to being used? Well, if you want to grow up a pure culture, you have to have a sterile media that you put the culture in to grow. I don't know why that's yellow and nothing else is. Usually when we have a plate, we will have a solid media because it's too easy for a liquid to be spilled out of a plate. If we have a liquid media, we normally grow it in a tube. However, that does not mean you can't have a solid media in a tube. And actually, we have two different types of tubes containing solid media. There's the deep, shown here, and that's just where you pour in the agar and the media and allow it to cool so it'll solidify and just go straight across. And then we have a slant, which actually you pour it in the same, but then you put the tube at an angle to cool. And then when it's cooled and solidified, you then stick it up and have the test tube, and then you have the slant. Any question about anything here?
Are those tubes similar to like what you would use for like swabbing an individual for some type of, let's say, like flu or something? Is that like the culture tube that essentially are used?
If you're putting the organism in a media, yeah, it would be a tube like this. Okay. If you're only doing the swab, like I've seen people do a swab and then they put it in an empty sterile tube. So it would be a similar tube, but obviously it doesn't have the media in it. Okay.
Yeah, makes sense.
Okay.
Thanks.
There are a variety of different media that we can use to grow bacteria. Today, we're only going to talk about general purpose media. It is a non-selective media, usually of complex composition, that supports the growth of many, but not all organisms. And it does not support the growth of a fastidious organism. A fastidious organism is one that is difficult to grow, and you have to add additives to get the growth to occur, like Streptococcus pyogenes, the bacteria that causes strep throat, would not grow on a general purpose media because it is a fastidious organism. And you have to add additives to the media to get it to grow. The easiest way to add the additives is you add red blood cells to the media, and then the red blood cells will supply the nutrients that Streptococcus pyogenes requires in order to grow. Some examples of general purpose media include nutrient agar with yeast extract, and it does have peptone and yeast and beef extracts in it. That's why this one has yeast extract in it. And that supplies many nutrients for many microbes to grow, but not the fastidious microbes. Another general purpose media would be tryptic soy agar, or TSA, and it is tryptic soy is obtained from soybeans. And that supplies the nutrients for the bacteria to grow. The nutrient agar is just a nutrient agar without the yeast extract added. So it's a little less rich than with nutrient agar with yeast extract. The point is that these are all general purpose media. They grow many microbes, and they are fairly cheap. Of all the media that we make, these are probably the cheapest, or at least one of the cheaper ones. Okay. Any question about general purpose media? We do have different type of media, and we're going to talk about that in a later lab. But for today, we're just talking about general purpose media. Whenever you're growing things in the microbiology lab, it must be appropriately labeled. Whoops, what did I do there? Everything should be labeled with your name, the date, the type of medium it is, like you can put NA for nutrient agar with yeast extract. You should have the inoculum. That's what you're growing. And I'm going to reverse this. The incubation temperature that you're growing the organism at is optional. You may put that on the plate. It's not required, but it frequently is included. Any question about any of that? All right. When you're labeling the Petri dish, you should write this information on the bottom of the Petri dish on the periphery. On a tube, you should label it on a piece of tape and then put the tape on the tube. Why we write on the bottom of a plate is because if the lid were to get separated from the plate, we want to know where this came from and what it is, whose it is, and what the bacteria are. And that's why we write it on a plate, on the bottom of the plate, because if it gets separated from the lid, then you don't know what it is, and there's no way to know. I once worked in a lab, and a lady had a whole tray full of hundreds of plates, and she had written all that information on the top of the plate. And then she tripped and spilled the plates on the floor, and all the lids came off. And so the whole experiment had to be repeated, containing hundreds of plates, because she had written her information on the lid. And so nobody knew what the results were for the experiment, what was the plate was, because the lid was separated from the bottom of the plate and the bacteria were on the bottom of the plate. Any question about any of that? So there is the periphery of the bottom of the plate. And there's the person's name, what the organism is, the date that this was done, the media, and then the temperature is optional. It may be included. All right, any question about that? Why you don't write in this area here is if you wrote here, then it would interfere with your ability to see the microorganisms in this region of the plate. Inoculation is the placement or transfer of microbes into a sterile culture medium. So we inoculate our culture medium with the organism we want to grow. And then how you make sure it's sterile is you sterilize your instruments. The incubation is the placement of the media in an appropriate physical environment for a period of time for the microbes to grow. Usually, we grow our microbes in an incubator. That's a device which warms up the culture and then provides humidity to aid in the microorganism's growth. You can have more complicated incubators that can control the amount of oxygen, the amount of carbon dioxide, as well as the temperature, the humidity, and then the time. So we have more simple incubators. They only control the time. You decide how long to grow it. It will control the humidity and then the temperature. And I'll show you an incubator in just a minute. Let me highlight that. Come on. Right here is an incubator that has a lot of dials and knobs, and that's to control the amount of oxygen coming in as well as perhaps the amount of CO2. And here we have a tray of water, and that adds humidity. That really is there to prevent the plates and the dishes from drying out quicker. And that way the plate can be kept in the incubator for a longer period of time, with the cells being kept alive for a longer period of time. And then it heats up the culture to whatever temperature you want. We have them set at either 30 degrees Celsius or 37 degrees Celsius. Let's see, anything else? Remember, when you're putting your plates in an incubator, you should put the plate in upside down. So let me make this a plate. You write your information on the bottom of the plate, and then you put the plate in the incubator upside down. The reason why is the incubator heats up the plate, and there's agar on the bottom of the plate containing moisture, and the water will come out of the agar and then condense on the top of the plate, forming a drop. And once it's a drop, it'll then drop down on the plate, which does not necessarily contaminate the plate, meaning the water is sterile, the top of the lid is sterile. The only time it would be contaminating the plate is if the water comes over here to the edge of the plate, and then you lift off the lid, and then the water drips down the edge and then drops onto the plate. That would contaminate the plate, because as you can sort of see, the lid is bigger than the bottom of the plate, and that's to keep the bottom sterile. And so this region of the plate would not, of the lid, would not be sterile. And so if the water comes down here, it would not be sterile in this region here, and you could contaminate the plate. But the main reason isn't because of contamination. It's because if the water drips down onto your organisms on the plate, you'll then get a mess of your organisms on the plate. So if you're streaking out the plate to get isolated colonies, if the water drops down, you no longer have isolated colonies. So we put things in the incubator upside down. So now the agar is on the top. And if water were to come out of the agar, it would form on the agar, and the agar would wick the agar around, so it would never form a drop. And even if it were to drop on the lid, who cares? We don't have microorganisms growing on the lid. Any question about any of them? All right. Under optimal environmental conditions, on an agar plate, we'd see visible bumps growing on the agar. These visible bumps we call a colony. You can actually look at bacterial colonies and generally tell them apart from fungal colonies by their appearance. There are some exceptions, meaning there are probably less than 5% of the bacterial colonies that look like fungal colonies, and less than 5% of the fungal colonies that look like bacteria colonies. But if we come down here and look at a picture here, you can notice that this is a bacterial colony, that's a bacterial colony, that's a bacterial colony, that's a bacterial colony, that's a bacterial colony, that is a fungal colony, and that's a fungal colony. What's different between the bacterial colonies and the fungal colonies? Name a difference.
The bacteria is more isolated.
more isolated. This one is isolated and that one's isolated. Meaning there's no other colonies around this one. So unless you mean something else, I'm not sure why you're saying that this one is more isolated than that one.
They're more uniform in shape.
Yeah, they're more uniform in shape, and that is a bacterial colony is more distinct, has a more distinct and clear, and in this case, roundish edge, whereas the bacteria doesn't have a well-defined edge. It kind of looks hairy or fuzzy. And that's generally how you can tell a bacteria colony from a fungal colony. You could also look at the size. These are the two biggest colonies on the plate. And that's because fungi tend to grow quicker than bacteria. That's not the best way to do it, because if the fungal colony were to start later than the bacteria, then it would be the same size or even smaller than the bacteria. All right, so when we're talking about colonies, you should realize that each colony is made up of 10 to hundreds of millions of cells. Each colony may have been a descendant of one cell or a clump of cells that was placed there during inoculation. Oops, didn't mean to underline that, meant to highlight that. So when we're streaking out this plate here, what we did, maybe streaked it out that way. Actually, it looks more like they streaked it out this way and then this way. What they did was, then they streaked it in this region here and this region here, here and here. They put down individual cells or individual clumps, which then grew in to form an isolated colony. Right here, we don't have isolated colonies. They're growing together. The same there. Any question about any of that? And this may have been formed by one cell, by two cells, or a clump of cells. We don't really know. That one's a little bit larger than most of the others, so I'll bet that was formed by a clump. Where was I? But the point is that the millions of cells in a colony were derived from one or a few cells that went through division, and bacteria grow by binary fission. We'll talk more about binary fission later in the class. Essentially, one cell will split into two in binary fission. All right, there's a little video for you to watch on binary fission. And I have a feeling this will be slight, so let's go ahead and watch that. Well, I can't get it to load. Only 16 seconds long.
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in two. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
All right, any question about that? You should note that when you're growing bacteria in a broth culture, you cannot see colonies. They will not grow in colonies. The cells will disperse themselves in the broth. And so all the cells will mix up. So colonies are not visible in a broth culture. We'll talk more about that later. although it might be in this lab, I don't remember. All right, when you're looking at colonies, you can get some colony characteristics. That is the way that the colony looks. So often there are visible colony morphological variations between different species of bacteria. Some different colony characteristics would be colonies can have different shapes. You can have a circular colony, a rhizoid colony, a regular colony, a filamentous colony, or a spindle-shaped colony. You can have a different edge of the colony, and we call the edge of the colony the margin. You can have an entire margin, and a circular colony has an entire margin. You can have an undulate margin, a lobate margin, or a curled margin. And if ever I ask you to describe the margin of a colony, I would expect you to be able to distinguish, is it entire? But to distinguish between undulate, lobate, and curled, I would never ask you to do that. Okay? It's just too difficult. The same thing with rhizoid and filamentous. I would expect you to say rhizoid and filamentous is different from entire or undulate, but I would never ask you, tell me, is this a rhizoid colony or a filamentous? Because they're just too similar. With elevation, and I'm unlikely to ask you what is the elevation of the colony, you can have a flat colony, a raised colony, and that's like a mesa, a convex colony, a pulvinate colony, and that's a superconvex colony, and an umbonate colony. Once again, I would never ask you to distinguish between a convex colony and a pulvinate. I don't know if I could distinguish those two. Okay. I'm unlikely to ask you about the elevation because to get the elevation of a colony, you really need to take off the lid and then lift the plate up to your eye level and then look at the colony from the side to determine is it flat, raised, convex, pulvinate, or umbonate. For the size of the colony, you can have punctiform, you can have small, moderate, and large. Anything that's a large colony is larger than the eraser on a pencil. Anything that's moderate in size in a colony is between the pencil eraser and the pencil point. That would be a moderate colony. Something that's small would be about the size of a pencil point. And then something that's punctiform will be just a pinprick, meaning smaller than the point of a pencil. And we're talking about a dull pencil, not a real sharp pencil. Any question about size? You can also distinguish the texture of the colony. Does the colony look smooth or does the colony look rough? For some reason, which I'm not sure why, students have trouble distinguishing between a smooth colony and a rough colony. You can look at the appearance of the colony. Is the colony shiny, which we call glistening, or is it dull? And then you can talk about whether the colony is pigmented or not. If the colony has any color to it, like purple, red, or yellow, we call it pigmented. If it doesn't have any color to it, it's non-pigmented. And that includes cream, tan, and white colors, also off-white. And then there's the optical property of the colony. If the colony is opaque, you can't see through it, and that's what most colonies are. There are some colonies which are translucent, and you can see through the colony, but you can't see through it well. There are some colonies that are translucent. And then there are colonies that are transparent, and they look like a drop of water. Very few colonies are transparent. Any question about any of that? I think I've got a picture here. Someplace in here, I've got a translucent colony. I'm not seeing it now, but this would be an opaque colony. That one there and that one there, these are translucent. And that one's opaque, that one's opaque, that one's opaque. Any question about that? So most of them are opaque. Like I said, that one's clearly translucent. There are no transparent colonies here. As I stated, transparent colonies are rare. Colony A is a large colony. I wouldn't even call that pale yellow, but it has a little bit of color to it. I wouldn't call that circular shape either. I'd call that more irregular. Shiny, that's hard to tell there. Pulvinate, I can't tell that at all. An entire edge, that I can see. If you look at this from here, I cannot tell which of these colonies are flat, which are mesas, and which are pulvinate. I just can't tell that looking here. Colony B is a medium colony. It's cream in color, irregular in shape, that I can see. It's a rough colony. It's definitely not smooth, nor is it shiny. And I can't tell you if it's flat or not, but they're saying it's flat. Any question about any of that? All right, when we're talking about colonies, it's important to understand what colony abundance is and what colony diversity is. Colony abundance is simply the total number of colonies on an agar plate. So to get the colony abundance, all you do is you count all the colonies. So here, there's too many to count. And it would be very hard to count them here. But in this region there, so we're talking about the colony abundance here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So there's 13 or 14 colonies in this region of the plate here. So that would be the abundance. The diversity would be the different types of colonies. And we can see that all of these colonies are together, they're the same, and that's probably the same. These are the same, these are the same. That one is clearly different. You know, I didn't see, there might be a colony there too. That one might be different than that. So here I would say there's at least 2 different colonies for the diversity, and there might be 3. So the diversity is either 2 or 3 for this region of the plate here. Any question about that? So here's another example. In this image here, what is the abundance of wild cats in this picture? Okay, think about it. And then you can also think about what is the diversity of the cat species in this picture. And since nobody's going to answer, I'm not going to tell you whether they're right or not. All right, so know the terms of this lab. There's a neat picture of somebody's hand. You don't need to look at the references. And now let's go to the worksheet. So in this lab, you're not going to be doing any of the lab. We're going to provide the data for you, but you do have to understand the lab. So in lab module 1, we have the ubiquity of microorganisms. We sampled a large number of environments, 1 through 5 here. Oh, these are the different protocols. Sorry, for procedure 1, procedure 4, procedure 5. Yeah, all right. Anyway, procedure 1, you don't need to answer this question. I should really pull out the question there. But procedure 1, we're answering what's in the air you breathe. So what we do is we open a nutrient agar plate, expose it to the air for a amount of time, and you need at least 10 minutes in the lab, maybe longer, because our air in the lab is filtered and it's fairly sterile. It isn't sterile, but it's fairly clean compared to most other places in the school or other rooms. And then you incubate it for 24 to 48 hours at 30 degrees. And it's asking you, what is the colony abundance on this plate? So answer that there. And then what's the colony diversity? Remember, with diversity, you have to determine what is a different colony species on the colony characteristics. Like these two are definitely different because they have different coloration. This one is definitely different because it has a different texture and margin compared to that one. And this one is circular, that's irregular too, as well as the size difference. There is a real small one, and there's a real small one, and there's a real small one. I'm going to assume that these small ones might be the same colony because they look pretty similar.
Question, are you looking for like a number answer, or are you wanting us to like describe, okay, well, I see this type of colony, I see this type of colony. What's I guess the
For a colony abundance, you just give the total number of colonies on this plate.
okay.
For diversity, you give the different number, the different number of different colonies on this plate. And as long as you're close, I will accept your answer. If you're not close, I will tell you this is wrong, try again. Okay? All right. And then procedure 1C is a thinking question. It is addressed somewhat in this lab, but you do have to think a little bit. And that is, what step would you take to reduce airborne contamination on a sterile plate that you are inoculating? So don't tell me something you can do before you inoculate the plate or something you can do after you inoculate the plate. You have to tell me something you can do while you are inoculating the plate. Okay? So when you're inoculating the plate, let's say you're streaking something. Can you guys see this? I'm trying to give you
Yeah, when you keep it in front of your body, or else the background kind of blurs it.
the background blurs it. Do you want
Well, I mean,
the background makes it show up. That's what normally happens. Anyways, when you're streaking the plate, there's something you can do to prevent microbes from falling into the plate from the air. And I will try to demonstrate one thing you can do, and it is mentioned in the lab, and that is you streak it like this. And the other thing I did show you, but it's not terribly obvious what the second thing is. You only need one thing, but don't tell me something like, make sure the agar is sterile before use, and make sure you put it in the incubator upside down, because that's not something you can do while you are inoculating the plate. All right, procedure 2. What gets on your arms when you put your arms on the lab bench? You can read through the procedure. To save time, I'm not going to read through it. The first question, procedure 2A, and I'll just abbreviate that P2A. Did table disinfection work? Here's a streak of the table before disinfecting the table, and there's a streak after disinfecting the table. So answer yes or no, and then answer the question, what is the purpose of allowing the table disinfectant to air dry? I don't think this is directly discussed in the lab, but it's pretty obvious when you think about it. So once again, it's a thinking question. And I think if you don't know, you can search online and simply ask, why do we allow the disinfectant to air dry? And I think you'll get the answer if you do a search online. Procedure 3, what's on your hands? Here is a picture of your hands before you wash your hands. And there's a picture after you wash your hands. Now, obviously, we're comparing skin that is a similar dryness. Like if you touch the plate with a dry hand, you need to wash your hands and then remove the water by drying your hands and then touching the plate. The reason being is if you were to wash your hands and then have water on your hands and then touch the plate, the water will wick the bacteria from your hands onto the plate. And occasionally I had students who did that and they got more bacteria on the plate after they washed their hands. And then I discovered they hadn't dried their hands, and that's the reason why. Okay. But that's not what's going on here. We have similar dryness of their hands, and you can see there is more microbes on your hands before you wash your hands. But after you wash your hands, there are microbes on your hands. It is reduced, but it's not totally reduced. And then you can answer, does hand washing work? And you should probably explain that. And then 2, suggest one reason why hand washing did not result in the complete removal of microbes. And it may have something to do with the normal microbiota of your skin. If you obtain an answer from your research, like online, provide a brief citation. This is a thinking question. It's not directly discussed in the lab, but you can get it. Excuse me. You can get it if you do research, but it's fairly difficult. It's much easier actually to get it from thinking than to do it from doing research. You get part of it from research pretty easy, but the entirety of it will be difficult to find it under research, but you can find it if you look hard enough. And like I said, it's easier just to get by thinking about it. Procedure 4, when you kiss your significant other, and we're talking about French kissing, I guess, what exactly are you giving him or her? What's on your lips and what's on your tongue? Here is a picture of the lips. Here's a picture of the tongue. And you should note that I put a note above these pictures. And then it asks A, which plate has higher colony abundance, the lips or the tongue? And B, why do you think this is the case? That is, why might there be more colonies in one plate than the other plate, or one plate than the other plate? Okay. You need to have 2 reasons for why. And there's actually at least 3 easy reasons for why. And actually, I've had some creative students come up with potentially 5 different reasons. Okay, but you must give an answer, 2 reasons why there's more colonies on one plate than the other. Any question about that? And then C, why was the tongue plate incubated at a higher temperature than the lip plate? If you look at the procedure here, the lip plate was incubated at 30 degrees and the tongue plate was incubated at 37 degrees Celsius. Procedure 5. Are there microorganisms in the soil? You can read through the procedure. And then we're showing you a plate. And then it asks, what is the colony abundance? Is it even possible to accurately explain this answer? Explain your answer. And then procedure 5B, is there greater abundance of colonies from the soil compared to the lip plates? Okay. And I'm going to warn you, some people only appear to look at the larger colonies on the soil, like this one and that one and that one and that one and that one. And they're not looking at the small colonies here and here and here and here. And then they're only looking at the small colonies here. There are some large colonies on the lip plate too. So both plates have both large and small colonies. I'm going to tell you that. And then answer procedure 5B. And then when you're done, submit it. This must be submitted by 11:59 p.m. this Saturday. Any questions about this lab? All right, if there's no questions, boy, my Zoom session might be having trouble. Is anyone there?
Yes.
All right, good. I was afraid my Zoom wasn't working. All right, so that's it. I'll be here until 8 o'clock, or the last student logs out to help you with the lab or answer any questions. Oh, if you have a question, you should log in early and answer the question, because students tend to log out. And once the last student's logged out, I'll log out also.